Hello, my name is Mason Cipollini, joined by Colin Brown and Leif Hilding. This is the final lab for OPTI 471B.
The human eye is a complex optical imaging system made of several important structures. Light first passes through the cornea, which provides most of the eye's optical power and also acts as a protective barrier against dust and small particles. Behind it is the aqueous humor, a transparent fluid that helps maintain the eye's shape while nourishing the cornea and lens. Next is the lens, the final major optical element. Unlike the cornea, the lens can change shape using surrounding muscles. This changes its optical power, allowing the eye to focus on near or distant objects. Behind the lens is the vitreous humor, a transparent gel that helps maintain the spherical shape of the eye and keeps the retina in place. Finally, light reaches the retina, the eye's detector. The retina contains rods and cones, which convert light into electrical signals sent to the brain. Rods are more abundant and highly sensitive, making them important for low-light grayscale vision. Cones are responsible for color vision are commonly grouped as red, green, and blue sensitive cones. Overall, the eye combines optical focusing, protection, and detection into one system. In this experiment, we will quantify different measures of visual acuity in our own eyes. Our first test was to determine eye dominance. Since our eyes are slightly separated, each eye sees the world from a different perspective. When switching between eyes, objects appear to shift position. With both eyes open, the brain tends to rely more on one eye's view. This is the dominant eye. To test this, we held a piece of paper with a small hole in it and viewed an object through the opening with both eyes. Then, we closed each eye one at a time. If the object stayed centered when one eye remained open, that eye was dominant. From this test, Mason and I were right eye dominant, while Colin appeared to have no single dominant eye. Next, we tested visual acuity using a Snellen chart. In this test, the observer stands 20 feet away from the chart and reads rows of letters that get smaller from top to bottom. The goal is to read the smallest line possible. Snellen results are written as 20 over something. The first line is the testing distance, while the second number is the distance at which a standard observer could read that same line. For example, 2020 is considered standard vision, while 2015 means better than standard acuity. Our results varied. Colin measured 2100, Mason had standard vision at 2020, and I measured slightly above average at 2015. Another test we performed was the Functional Acuity Contrast Test, or FACT. This test measures contrast sensitivity and resolution, in other words, how well we can distinguish closely spaced details at different contrast levels. The chart contains sinusoidal patterns that increase in spatial frequency from left to right, measured in cycles per degree. As you move down the chart, the contrast decreases, making the patterns harder to see. We conducted the test from a distance of 1 meter.
Next, we took a look at our blind spots. The blind spot is a small gap in the visual field of each eye due to a lack of photoreceptive cells where the optic nerve and blood vessels attach to the retina. First, we found the angular size of the blind spot. This was done by closing one eye, then looking at a card with a circular cutout in it. We then moved the card so that the cutout was in the blind spot, where we could no longer see the hole. Then we brought it as close to the eye as possible while still remaining completely in the blind spot. Measuring the distance and size, we calculated the angular extent of the blind spot at around 5 degrees. Next, we found the location within our field of view. This image was put on a screen, then we closed the right eye and looked at the cross, adjusting distance so that the dot was invisible and within the blind spot. We got a range of 12.8 to 16 degrees temporally from the center of vision. Next, we tested our flicker fusion frequency. This was done by driving a green LED at several voltages, then finding the frequency at which a driving square wave made the LED appear to be a single solid color instead of a flashing light. We found that the driving voltage, increasing brightness, correlated with slightly higher fusion frequencies, and we also found that the fusion frequency was higher outside of the fovea. Then we looked at the spectral range of the human eye, finding the shortest and longest wavelengths detectable. We had a lot of issues with the equipment. We were unable to get a clean output frequency, as looking into the instrument one could see a spectrum of wavelengths. Another potential issue is the calibration of the monochrometer, where the values reported for the wavelength don't seem accurate. We found a shortest wavelength of 389 nanometers and longest of 683 nanometers.
We then tested our color vision using Ishihara test plates, which place a shape or number into a collection of dots. This array consists of certain color combinations to test for certain deficiencies. This is because color blindness is primarily caused by a deficiency in the short, medium, and/or long-wave pathways in the eye's cones. As you can see, I have said deficiency and am classified as colorblind. We also looked at some optical illusions, which are meant to manipulate the electric signal going to the brain's cortex from the retina, tricking the mind into misinterpreting certain details. For example, you could see this pattern in which I and others saw a sort of light spinning within the rings, as well as this elephant which has legs that don't properly match up. Additionally, the patterns in the book seen on the right can appear to be 3D if you view it a certain way. Finally, we were able to use some virtual reality headsets, which interacts with every part of the eye for overall immersion. This allowed us to feel like we might actually be boxing in the virtual ring or really flying the virtual remote control plane. Overall, this lab provided a great opportunity to understand how each part of the eye works and how certain details can impact the way we see the things around us.